So this is kind of like early development of mammals. And so following from the last step of fertilization, the first thing that happens is the pronuclei fuse, so the two 1L or 1N nuclei of both the egg and the sperm fuse together, and once that happens, the second polar body forms, or yeah, around there is when the second body forms. So then what happens is they separate into two cells, and then those two cells become 4 cells. Those 4 cells become 8 cells. Then at the 8 cell stage, it goes into a condensation stage, and those 8 cells kind of like become one undefined mass. And then the next stage is when it becomes a blastocyst. And what happens is the cells, there's a lot of cells at that point, and there's 2 definite regions. One is called the trophoblast, and the other is called the epibody. And it's kind of like, if you imagine like a shell, and with like a mass on the inside. The shell is the trophoblast, and the thing on the inside is the epibody. And all during these stages, they're still covered in the zona pellucida. Okay. And that's actually really important because all during this stage, it's traveling through the fallopian tube, and it's still protecting the embryo from anything inside the uterus. And it's also preventing it from implanting too early, and which is a problem. There's certain medical conditions where the embryo will shed the zona pellucida too early, and it will implant inside the fallopian tube, and then a child will begin developing
ectopically.
Ectopically inside the fallopian tube, which is really bad. So once it reaches the uterus, the embryo sheds the zona pellucida, and a process called implantation occurs, and that's when the embryo gets on the uteral wall, and then more steps happen where the placenta forms and there's further differentiation. The thing that is the trophoblast contributes to the placenta, and the inner cell mass or epibody is what contributes to the actual organism.
Sweet.